Welcome to DCRI Research Conference. Before we get started, wanted to remind everyone that this is being simulcast and recorded. So those people in the room, if you have questions, speak up so the mics in the ceiling can pick them up. Happy to introduce our speaker today. Jeff Ginsburg is here. He's the director for the Center for Applied Genomics and Precision Medicine and is the founding director of that center. He's been at Duke since 2004 as a professor of medicine, pathology, and biomedical engineering. His research is addressing the challenges for translating genomic information into medical practice using some new and innovative paradigms, which I suspect we'll hear somewhat about today, and in the integration of precision medicine into healthcare. He's internationally recognized expert, has been on many panels and commissions around the world, has over 200 papers in the field, and has funding from many agencies and foundations as well as industry. Prior to being at Duke, he was at Millennium Pharmaceuticals. I did notice when I was reading his CV that he has 15 papers currently listed as submitted, which there's no grass growing under his feet. So welcome, Dr. Ginsburg.
Thank you, Doc. And it's great to be here, and I'm told that you need to pay attention to this slide more than anything else. So it is an honor to have a chance to talk to you about something that I'm very passionate about, precision medicine, but also something that I think will affect not only just me and you, our children, our families, and our friends in our lifetimes. I hope I can convince you of that today. These are some of my disclosures, but probably more importantly, these are among some of the colleagues and collaborators that have worked with us in our center, but also across the campus in the Pratt School of Engineering, as well as at other institutions such as the University of Michigan and University of Virginia. So this journey began, at least in my mind, on April 14, 2003, when I think by some acclaim we achieved what was thought to be a moonshot of sequencing the first human genome, a project that was conceived in a snowstorm in Alta, Utah in the 1980s by some of the world's top geneticists who were snowed in and had a chance to have a conversation about what they would wish the field could do. And miraculously it was achieved. And Francis Collins at that time said that this was not the end, but really the beginning of an era of genome information, genome biology applied to health and medicine, and encouraged all of us to be a part of that. Now, in the intervening years between now and then, you may remember that when he was senator, President Obama submitted a bill before Congress called the Genomics and Personalized Medicine Act, which you can't read from the back of the room, but it says to improve access to and appropriate utilization of valid, reliable, and accurate molecular genetic tools by all populations, and thus helping to secure the promise of personalized medicine for all Americans. So we actually had a hand in helping Senator Obama write that bill, which was never passed by Senate, but it gives you a sense of his vision, which he re-articulated just a couple of months ago in the State of the Union address when he announced the United States Precision Medicine Initiative, which has some of these aspirational goals to really begin the foundations for curing chronic diseases and bringing novel therapeutics to the marketplace. You can see him here at the White House, flanked by a model of the DNA double helix, which came from the NIH for that particular meeting. So I think we're at really a really interesting time, the convergence of genome biology, genome sciences, and medicine. But it's worth really thinking back to where the field began, and I think when I talk to people about precision medicine, particularly clinicians, they feel that they've been practicing precision or personalized medicine for all of their careers. In 1961 was the first time the term factor of risk or risk factor was used in the medical literature, coming out of the early findings of the Framingham Heart Study, which of course brought us an opportunity to risk stratify individuals on the basis of both clinical as well as laboratory and historical data. I think that's really what we're talking about when we talk about being more precise in medical care, is to put people on a more refined continuum of understanding their their propensity for disease. And in the 50 years since that publication or so, I think it's arguable that there have been some radical transitions in the way we think about biology and science, starting with the notion that 50 years ago that we could really only describe disease on the basis of its anatomic location or histologic appearance. But then in the 1970s and 1980s, with the advent of some of the molecular tools of molecular biology and recombinant DNA technologies, we could begin to think about an analog view of biology where proteins were turned on and turned off and could be used to describe disease paradigms. And I think we can probably all agree that with the ability to assay an entire genome worth of data, that we're now in an era where we can really describe disease on the basis of a series of digital phenotypes. So the Genome Project did not just give us a sequence of a human genome, but it gave us a toolbox of technologies that can help us redefine a number of medical paradigms. So you can see we have now 3 billion base pairs with millions of single nucleotide variants, the ability to assay an entire transcriptome either by chip technology or sequencing technology, the proteome, which with the splice variation and post-translational modifications might give us several 100,000 of protein entities and small molecule metabolites constituting the metabolome. This constellation of tools is what I guess Craig Venter has called the software of life. And the way we have tended to use it, particularly in our views here, is that we might be able to aggregate this data and not only consider what is more the traditional venue for medical predictions based on clinical, environmental, and other factors, but to incorporate the wide variety of molecular technologies we just saw, and as well as imaging and perhaps electronic health data to build models and signatures that would refine our abilities to predict who is at risk for developing disease, who might be more likely to respond or have an adverse response to a drug, as well as how we respond to environmental stimuli. And I think we can begin to see how these tools could be used across the entire continuum, as some might say, from womb to tomb, when you think about prenatal testing and reproductive testing that is currently available, and of course across the lifespan now, this being the course of a depiction of what a chronic disease might look like as it evolves over time, being essentially asymptomatic or pre-symptomatic until it reaches some clinical horizon, presents to medical care. This is where most of medicine is practiced. At the time where the cost of care is the highest, the disease burden is highest, and when the opportunity for reversibility is the lowest. And I think we can begin, or I hope as this talk goes on, you'll be able to see how the deployment of a number of these novel tools and technologies now gives us an ability to really detect who is at risk at the earliest possible time in one's lifespan, to refine our ability to screen for those diseases in the highest risk individuals molecularly, and to use these tools for better diagnostics, prognostics, and selections of therapy. When I came to Duke 10 years ago, this was really the problem that I thought we could all collectively begin to try to solve. How do we take something that looks like a genome sequence packed full of potentially important information for the care of an individual, or potentially for the care of a population, and realize this vision that I just showed you of achieving more refined and personalized health care? And these are the 6 areas that I'd like to try to cover today that might give us a handle on how we're progressing. I think the advent of genome sequencing, mobile and electronic health technologies, the engagement of our patients, our ability to take this information to define better therapies and diagnostics, our embracing the learning health system model, and more recently, the notion of applying big data and big data analytics to this problem. So let me begin with sequencing, and maybe I'll just ask if anybody in the room has actually had a chance to peer into your genome or had your genome sequenced or genotyped. A few. One. Including myself, too. I think this idea really began only about 10 years ago. The notion that we could actually use genetic information to associate with chronic diseases, with the first publication, the publication of the first genome-wide association study, this is a karyogram that shows this little lollipop-looking symbol on chromosome 1, and this was the identification of variation in the complement factor H gene associated with age-related macular degeneration. And the two groups that simultaneously published this result in Science magazine, one was from Duke, from the Center for Human Genetics, led by Peggy Vance, and the other was from the National Eye Institute. And what was spectacular about this is that no one even suspected the complement factor H or its molecular pathway was involved in age-related macular degeneration. And today there are clinical trials. There are molecules in the clinic that are aiming to modify this particular pathway as a way to mitigate or ameliorate that disease. So only 10 years later, based on a completely unbiased finding, there is now drugs in the pipeline targeting a pathway that had never been even thought about as being related to that disease. Now, fast forward 10 years later, there are now over 4,500 genome-wide findings across 300 complex phenotypes with over 2,000 or 3,000 publications that are now nicely cataloged both at the NHGRI and with a concomitant European organization. But what I think is absolutely stunning about this is that many of these gene variants have occurred in genes that were never really thought to be associated with the disease in question. So this unbiased approach, a genome-wide approach, has really given us some spectacular insights into biology that had never been suspected. And that has led to the kinds of activities that I just mentioned with respect to AMD and complement factor H modulation. But also it's been somewhat disappointing because many of us expected that having knowledge about having a risk allele would be clinically informative and would allow us to tell a patient that he or she is at higher risk for developing the disease, where in fact the odds ratios for a great many of these findings, certainly the vast majority, is that the odds ratios of carrying the risk allele are far less than 2, actually much closer to 1, 1.1, 1.2, 1.3. Odds ratios are certainly not something that would rise to the level of clinical importance. So I would argue that maybe we've overlooked something important, and that's family history. When we were in medical school, we were taught that we should take an adequate 3-generation family history in order to be able to give a patient an understanding of their risk and possibly even allow their relatives, their siblings, their parents to have a better understanding of familial risk. But in point of fact, if you look at electronic medical record data from places like Duke or other top-tier institutions, the presence of a 3-generation family history in the medical record is less than 1% of the records. This is a nice study by Nadim Qureshi and his group in England, which essentially showed that in a pragmatic, real-world clinical trial that was cluster randomized to give some practices a paper-based family history tool that allowed the patients to identify better their family histories of cardiovascular disease, that not surprisingly that those practices that had the intervention picked up the risk for CVD at a rate that was 40% greater than the control groups. And in an unexpected finding in this particular study, they found that those patients that recognized that they had a family history of cardiovascular disease tended to adopt lifestyle and behavioral patterns that were much more beneficial in changing their diet, exercise habits, etc. Now, we've also been interested in family history, and over the last 7 or 8 years, we've, with some help from the Department of Defense, we've developed a patient-facing, web-enabled, evidence-based, guideline-driven family history tool that is currently being integrated into Epic at Duke, as well as at 4 or 5 other health systems that were funded to do this with currently under NIH funding. And some of the early data that we've had from our family history program, which is called MITRI, that's the name of the tool, is that before practices and providers that didn't have access to family history data were, in many cases, ordering inappropriate screening tests for patients that were actually at low risk, that did not screening, or inappropriately screening people who they thought were at high risk for disease that subsequently were found not to be. So I think it's important to recognize that we can get a lot of important valuable information from something that's not particularly genomic, but has certainly genomic-like implications for risk. On the other hand, we cannot ignore that sequencing is here, and it's becoming more and more commoditized almost every day. And this slide just shows you that the Genome Project, completed 10 or more years ago, cost almost $3 billion to sequence a single genome and 13 years to do so. Whereas because of the investment in technology development, it's now conceivable to have a genome sequenced in less than a day at less than $1,000. And as this picture indicates, to have a whole sequencing platform that essentially is on a USB port that can fit into a laptop computer, so you can stream data from the sequencer directly onto a hard drive. That is at least the proposal from Nanopore Technologies. And that has led to the ability to carry out mega studies when it comes to genomics. And these are just a few of the studies that are either on deck or are currently running today. The National Health Service in the UK is sequencing 100,000 genomes. It's linking to their electronic medical record. Saudi Arabia has partnered with Life Technologies to do the same thing with a strong emphasis in cardiovascular disease. Geisinger and Regeneron, Geisinger Health System, Regeneron Pharmaceuticals have also developed a partnership around sequencing 100,000 Geisinger genomes and linking it to their EPIC EMR. And Genome Qatar is also about to begin to sequence the 300,000 native Qatari genomes in that country and link it to their Cerner EMR system. So the hope is that not only will that information lead to better information for clinical care delivery, but as I'll show you, also potentially give us important information and insights into underlying disease biology. And you only need to be a student of the New England Journal of Medicine to recognize how sequencing has become potentially really important in the management of some patients. So this is more of an infectious disease-oriented series of studies, one in which the etiology of MRSA in a neonatal intensive care unit was tracked down by real-time sequencing across the entire unit and being able to identify the epicenter of this outbreak and quarantine that individual. The etiology of Middle East respiratory syndrome as a camel-to-man transmission was also documented by sequencing. And a young boy who had a fever of unknown origin and was in a coma had the diagnosis of leptospirosis made by sequencing his CSF, and he received a life-saving antimicrobial therapy on the basis of doing so. And in the non-infectious disease realm, we're seeing, for example, sequencing of feces in order to detect occult malignancies, becoming far superior technology to the traditional immunohistochemical measures for screening for colorectal cancer. And in this paper, now 18 months ago, was the first case series of 250 individuals that were sequenced because their children who were sequenced that had diagnostic dilemmas. And what was found, actually, interestingly, in this series was in over 25% of the cases, a diagnosis was made on the basis of sequencing, which ended the diagnostic odyssey for those patients and their families. And then roughly out of 130 that were sent to insurance companies for payers to reimburse for these, 125 said yes. So the reason being that the cost of doing a sequence has become so acceptable today that it actually makes it much more cost effective to fund sequencing versus continuing to do a lot of ancillary tests and a lot of ancillary diagnostic testing at cost. Now, to me, this is probably one of the biggest reasons to really consider genome sequencing as an important modality for diagnostic dilemmas. And maybe you've heard of the case of Nick Volker. It's now several years old, and as this indicates, the Milwaukee Sentinel that followed his care at the Medical College of Wisconsin won a Pulitzer Prize for writing his story, which he presented to physicians at the Medical College of Wisconsin with a failure to thrive syndrome. He had over 120 surgical procedures on his abdomen and for what looked like an inflammatory bowel disease like illness. And in consultation with the clinical team and of course the family, in sort of a Hail Mary notion, they decided, well, we should just sequence Nick's genome and see if we can find something that would end his suffering or make a diagnosis. And they actually did find a functional mutation in the X-linked inhibitor of apoptosis gene, which had actually never been seen with a phenotype such as this. It had been seen in lymphoproliferative disorders. And interestingly, it was a curative procedure for those individuals was a bone marrow transplant. So now we have a phenotype associated with, or a novel phenotype associated with a mutation that has a pretty dramatic therapy associated with it. And again, sort of the Hail Mary, the parents and the team decided to give Nick a progenitor cell transplant, and Nick was cured. He's now a healthy 9 or 10-year-old who eats normal diets and plays with kids his age, and his life was clearly saved by sequencing his genome. So to me, that's at least one case in point as to why we really need to embrace sequencing as a technology that's here today. Now, sequencing is not easy. We've solved many problems when it comes to sequencing genomes over the years, from the data generation problem to the data cleaning problem, even to beginning to think about what are the functional aspects of the genome. But as this slide indicates, the real bottleneck in bringing sequence information to you and me and to health systems is the interpretive bottleneck and the ability to deliver information just in time to practicing providers and the clinical team. One program that NHGRI is sponsoring called the Clinical Sequencing Exploratory Research Program has generated a one-page genome report. This comes from a program called MedSeq. It's at Partners Healthcare. Robert Green is the PI, and I think they've done a fantastic job in distilling at least what we understand today in terms of actionable variants from the genome into a one-page report that talks about the monogenic risks, the carrier status, pharmacogenomic potential for this particular patient, as well as a blood type, single-page report. So we're making progress, and hopefully we'll see genome reports such as this showing up in the Duke system sometime in the near future. So I'd like to then move on a little bit to this notion of the learning health system and why it's important to this whole field of precision medicine. I'll start really with a cancer example. And as I think many of you know that the cancer genomes exhibit a completely different set of variation than our germline genomes, and this is a really beautiful review by Bert Vogelstein, really summarizing the state of the art of about 140 genes that are driving the cancer phenotype today. They actually tend to revolve around known oncogenic stimulating pathways. And there are some issues that are raised in this review about genetic heterogeneity, that not every cell in a tumor is the same, and therefore there's opportunities to miss an appropriate diagnosis and match a therapy to a patient's tumor. And the oncologists are really in this unique position of not only having the tumor genome to use to make medical and treatment decisions, but also having the germline genome as the reference, so they actually have to look at two genomes in order to optimize a patient's care. Now that's the state today, but 25 years ago, as I said earlier, our description of disease was largely descriptive. So here's the way non-small cell lung cancer might have been viewed at that time: squamous, large cell, small cell adenocarcinoma. But today, thanks to our ability to sequence cancer genomes, we now have a series of activating mutations, particularly in adenocarcinomas, that have actually led to the development of targeted therapies that can actually that target that particular activating pathway. So we now have EGFR inhibitors, KRAS inhibitors, EM4 ALK inhibitors. And not only do we have this plethora of targeted therapies that are approved by the FDA, but also as some of these boxes indicate, there are drugs in development across many of the major pharmaceutical companies, such that today, if you had a lung cancer patient in front of you today, they can get their tumor sequenced, they might be a candidate for an FDA-approved drug, or they might be a candidate for referral to a clinical trial for any number of these therapies that match the underlying biology of their tumor. But here's one of the problems. And this is actually data that was obtained from the Dana-Farber Cancer Institute. So in their case series, greater than 50% of non-small cell lung cancers actually have one of these mutations that I just showed you. But if you go out into the community in North Carolina, or anywhere else in the United States, only 5% of cancer patients with a non-small cell lung cancer diagnosis are getting an EGFR measurement in the United States today. So we have a gap between the knowledge that we have about what could be done to target a therapy to an individual and what is actually being done in terms of the access and knowledge base of the community-based physicians in particular. So we have to figure out ways to bridge that gap. And I've argued that one of the solutions is to actually use the health system as a way to learn, so we can begin to drive information at the point of care, at the point of decision, and allow clinicians to understand, what tests to order for a patient with this particular diagnosis, and when those tests come back, what might be some of the treatment options on the basis of their molecular profile. So the architecture, in a simplistic view, would look something like this, where we have a healthcare delivery information technology system that, of course, has the electronic medical record data, that has patient sequence data that could be then compared to some genomic knowledge base or reference genome, and of course, as I've argued before, incorporating family history, that this would be one of the ideal ways to bring this data together at the point of decision making for providers and patients. The NIH has launched a series of programs to achieve some of this goal of implementation of genomics into healthcare systems, into electronic medical records, the Electronic Medical Record and Genomics Network, or the eMERGE network. IGNITE is implementing genomics into practice. I mentioned the Clinical Sequence Exploratory Research Group that has been working on that report that I showed you, the Pharmacogenomics Research Network, and the Global Genomic Medicine Collaborative are all organizations and networks that are really working towards solving some of these challenges. And I just wanted to highlight one, which was the eMERGE network, and you can see the sites that are participating in the eMERGE network. Note that there are none in North Carolina. I just would say that one of the tickets to entry into the eMERGE network was that you had to have an electronic medical record that was linked to a biorepository, which I think we're working hard at doing here, but we're not quite there yet. On the other hand, what eMERGE has been able to do by virtue of that linkage is to actually use electronic medical record phenotypes to do genome-wide association studies of the type that I showed you earlier. So electronic phenotyping, electronic consents, sorry about that, engagement of the community, thinking through the data security and data privacy issues, also beginning to work in the pediatric population on pharmacogenomics as well as other genetic risks, and eventually beginning to create a warehouse of clinical decision support tools that could be used to promote these goals. So this happens to be the clinical decision support clinical decision support engine that fires at Vanderbilt when a patient is prescribed clopidogrel. And the reason this can fire in real time is because Vanderbilt has preemptively genotyped most of their cardiovascular patients. So when a patient comes into Vanderbilt and gets prescribed a drug, there's an automatic search of the genotypic database that allows this rule to fire, and this just indicates that genetic testing has been performed on your patient, that this individual happens to have a variant that makes him or her a poor metabolizer of the drug, therefore recommending that an alternative dose or an alternative to this medication be given. And that's the kind of thing that will really, I think, help providers give them very pragmatic information at the time that they're making a decision to treat a patient. And that is at least some of the attributes of a learning health system that is going to enable delivery of precision medicine. I wanted to just remark about particularly how this field it needs to embrace or is being embraced by pharmaceutical companies. And this may be one of the most obvious reasons why this needs to happen. Maybe you've seen this slide in this room before, but this is a log linear plot of the productivity of the pharmaceutical industry in billions of dollars per R&D spend to yield a single product. And it's a log linear relationship that looks something like it will get close to zero within the next 10 years unless something dramatic changes. So, and it's been called Eroom's Law, which is Moore's Law spelled backwards by the authors. Sort of a joke, but maybe not really, because I think it represents a significant problem that needs to be addressed. And I'm not saying that genomics is a panacea, but certainly one can envision that if we did a better job of using genetic tools to do lead optimization, to do early preclinical safety and efficacy trials, and probably most importantly, to select the right patients to be in clinical trials, we might be able to address some of these issues. And this was demonstrated quite clearly more than almost 15 years ago when by the iconic trial that was run by Genentech to bring Herceptin to the marketplace, using the HER2/neu biomarker to select patients to receive breast cancer patients to receive Herceptin. So this is essentially what was used in the trial, 470 patients with a 50% response rate and 1.6 years of follow-up. But without that genomic marker, the trial would have required, as you can see, over 2,000 patients. The response rate would have been less than 10%, and it would have taken 10 years to complete. It would have never gotten done, and the drug would have never been approved. And there need to be more and more pharmaceutical companies that are thinking about how to redesign trials such that they can be much more selective on the basis of protein biomarkers, such as this case, or things that have a more genetic and genomic basis. And it's not like this has been happening. Most of this data that's now on the FDA biomarkers page is a result of actually academic work rather than pharmaceutical work on known drugs. When the genome was sequenced in 2003, there were only 4 FDA labels that contained any genetic or genomic information, and now, as you can see, there's over 150. The other place where there might be some opportunities is in the preclinical space, and I just wanted to mention the tissue chip program, which I think is one of the most innovative things I've seen in a long time coming out of NIH and NCATS in collaboration with DARPA and the FDA. But the idea here is essentially to create a chip that contains both neurovascular cells, a gut-like organoid, liver, kidney, heart and lung components, all that allow systematic and selective administration of therapeutics or changes in genetic background in order to really define in a rapid, high-throughput way potential efficacy and toxicity measures without the need for a model system or even a human being. And this what you're seeing here in the background is just an example of the kind of functional types of attributes that this chip has, and there's also similar types. This is a cardiovascular, these are cardiomyocytes that are actually beating rhythmically, and that which stress-response relationships can be measured, as well as a series of other measures. There are similar physiologic measures on kidney, as well as pulmonary and neurovascular tissue. And I think this is moving forward. Companies are beginning to use this in preclinical development. And one of the visions, I think, from particularly from the NIH and the groups that are working on this, is that, that we will potentially all have our own physiologic, genetically perfect chips for each one of us. So if you take our induced pluripotential stem cells from either you or me and begin to create these organoids, so I have, on my chip, I would have representations of my heart, my lungs, my gut, my neurovasculature. I could begin to actually profile how I would respond to a particular drug or not. And one can imagine the possibilities. So this is obviously way out there, but something that is currently being worked on at the NIH, by the NIH and others. And it's not like we haven't been successful, and I wanted to just make sure we didn't overlook some miraculous accomplishments by industry to bring targeted therapies to the market, many of which are in the area of cancer, such as crizotinib for non-small cell lung cancer with the EM4 ALK mutation, or vimorafenib for melanoma with the BRAF mutation, and others. And in the last couple of years, there's been tremendous progress made in chronic diseases like cystic fibrosis. So bringing ivacaftor to the market for individuals that have the G551D mutation in the cystic fibrosis transporter gene has changed the lives for 5 to 7% of patients with CF who had limited mortality and poor quality of life. Now they're really functional, better quality of life, and really have a chance to live well into adulthood, if not beyond. I think that's really remarkable. And if you were at the ACC last 2 weeks ago, you heard about some landmark trials for the new class of cholesterol-lowering drugs, PCSK9 inhibitors. 10 years ago, this publication is what started the whole thing off, was the observation that individuals who carried essentially activating mutations of the PCSK9 gene had observed markedly lower LDL cholesterol levels. So that having those mutations, having that genetic variant really was an important signal and clue that really enabled all the PCSK9 programs to move forward. Now, in terms of diagnostics, this is an area where we spend a lot of our time. The notion being now that when you have all these genome-wide technologies available to you, you can begin to assemble the data statistically to differentiate between two different biological states, let's say health and disease, or pick your phenotype. So these types of molecular signatures can be used not only for diagnosis, but for drug response, and also if you think about what these genes actually are in this heat map, begin to get some insights into the biology that underpins that response. And this has been taken advantage of not just recently, but for almost a decade. In cancer, the Oncotype DX RNA prognosis test was the first tests were done in 2006 or 2007, and now literally hundreds of thousands of women with breast cancer have been tested for prognosis, and a decision was made whether to give them adjuvant chemotherapy or tamoxifen on the basis of the result of this test. And there have been some copycat tests and other tests that have also, with a similar platform. In the cardiovascular disease arena, the tests that are available are mostly blood-based, but the AlloMap test has been used in some centers to monitor patients. This is following cardiac transplantation. This is an 11-gene RNA signature from whole blood that really gives insights as to what's happening in the myocardium without the need for an endomyocardial biopsy. And it's been an FDA-approved test for several years. The Cora CAD test, similarly, is a 23-gene blood RNA test that has direct correlation with the degree of obstructive coronary disease, and as depicted on this slide, where the test score is directly proportional to the number the degree of global stenosis across the coronary beds. And I just want to highlight this particular test because it was actually the basis and the foundational science for developing this test was done in part here through the CathGen registry, having the foresight from Chris and Kristen to collect RNA samples from every patient that were undergoing a cardiac catheterization, and then perhaps a leap of faith, but we were able to show with Bill Krause and others that there indeed was a correlation between RNA profiles and a degree of coronary obstruction, which really gave this test, which is now on the market, its likelihood of success. In our own work, in our center, we tend to think about exposures as a way to do perturbations on human subjects and to look at what some call the exposome, which is maybe the sum total of the kinds of exposures we will have throughout the course of our life: radiation, stress, lifestyle, infections, drugs, diet, pollution. And we've added this Exposomics descriptor to the fact that we can measure our host response, how we as respond to these environmental perturbations, and use as a proxy for this the expressed genome, and looking at these types of things over time. Now tomorrow I'm going to be giving a talk at the Cardiovascular Research Center on this work, so I'm not going to go into it in any really great detail, but I just wanted to highlight why it's important. And this is a case about 2 or 3 years ago from Kville. The night before the UNC game, one of the students developed fevers, a headache. She was tenting. She went to student health. She was diagnosed with a viral syndrome, went back to her dorm. Her astute roommates noticed that she was becoming confused and confabulating and took her to the ER, where I'm told, actually one of the cardiology fellow on call made the diagnosis of bacterial meningitis and actually got her appropriate treatment, and she survived. But this is a case where if she had not survived, this could have been the result, where she would have been eventually, had fulminant sepsis and died. So it really indicates the importance of being able to make a accurate diagnosis of viral versus respiratory disease. This has been the basis of a lot of our work, and as I said, I'm not going to go into the details, but we have now a portfolio of gene signatures that are based on the host response that can really differentiate between viral infection, bacterial infection, and even fungal infection. And we're bringing those things forward into the clinic such that we can now present to a primary care physician or even an ED or student health doc something that might look like this, where we can help them understand whether this is a viral infection alone, bacterial infection, a non-infectious fever, or co-infection, all of which would have very different therapeutic implications, of course. So let me move on to this notion of patient engagement, which is obviously patient-centeredness and patient engagement is obviously one of the hallmarks of the way we think about medical practice today. From the point of view of genomics and precision medicine, this really began around the time that the genome was sequenced, with the advent of a number of different organizations aiming to help to use genomic tools and their own personal phenotypic information to drive to better research and better clinical outcomes. So there's the Personal Genome Project, which George Church started to originally sequence 10, now close to 10,000 individuals that make their genomic and phenotypic information completely public. Do-it-yourself genomics. You've heard of 23andMe, the direct-to-consumer, originally direct-to-consumer, now pharmaceutical partner of a lot of a burgeoning pharmaceutical partner using, again, personal genomic information and genotypic information. Iodine is a patient-reported website around drug and drug responses, aiming to highlight what drug profile, what individual profiles might lead to better drug responses in individuals. So my point is that we're all now surrounded by this incredible sea of information. We have our health records. We may track ourselves. We obviously can get our DNA sequence. We also may access social media sites, subscribe to surveys, use cell phones, and so on. And we just need to be thinking about where we want our data to be used and how should we share them amongst ourselves, share it with our caregivers, give it to clinical trialists and researchers, and all of this could be really quite compelling and powerful. And let me show you an example of this. I call this is where Moore's law and Metcalfe's law sort of converge. So Moore's law, as you know from the computer industry, says that the compute power of a chip will double every 18 months. And you can see that law expressed here sort of in comparison to what's happened to, this is actually a cost plot of Moore's law. And this is what's happened to the cost of sequencing a genome. So you can see that sequencing genomes has outpaced Moore's law significantly. Metcalfe's law talks about the social network, and the power of the social network as being the square of the number of users in a social network. And where I see elements of convergence of these is in studies like this, where 23andMe was able to use their social network of self-reported data, which at the time they were calling 23andWe, to link that to the genotypic information that they had already gotten from their direct-to-consumer programs, and completely replicate genome-wide association study results that were done by more rigorous clinical studies before this. So self-reported data, genotypic information, really giving us a handle on the root causes of disease using genomics. And we've also taken a stand a little bit to move this field forward. We started a new magazine. It's called Genome. It's meant for the public. It's meant to bring this information to reality for lay people as well as patients. And I have a few copies of the latest issue up here if you'd like to see it. But it really is meant to fill a gap, and it's free, by the way. Any of you can subscribe. In terms of really getting the patients to be more engaged, the public to be more engaged in both genomic research, but also to demystify what genetics and genomics might do for them clinically. I wanted to just touch a little bit on mHealth and eHealth. I think several of you in the room are probably either researchers or subscribers to this. If you read the Harvard Business Review in the fall, the prediction is that there'll be 20 trillion devices that will be somehow connected to one another over the internet. The Internet of Things is what some people call it. But a large number of these are these kinds of wearable devices. So you can have monitors in your shoe, around your wrist, on your waist, around your neck. Many of these are continuously tracking your data for you. But I would argue that this is, for the first time, we have an ability to get continuous phenotypes of the type that we've never had before. You know, in a clinical study of the type that many of you do, the only time you get a phenotype is when the patient shows up to the clinic for evaluation. But large dead spaces in between exist, and to me, these types of technologies are potentially allow us to see what's happening in between the standard clinical visits. And of course, I think I got this slide from Rob, but the data on cell phone usage is pretty impressive, with over 7 billion cell phone accounts around the globe, and many of the phones are being used to deliver important health messages to underserved and other types of individuals for prevention. And of course, in the last few months or so, we've now seen the emergence of HealthKit from Apple, and more recently ResearchKit, again allowing refined data tracking over long periods of time. And it's kind of wondered why this is called a phone, but that's probably a topic for another day. You've also perhaps seen that Google has invested in a contact lens that can sense and wirelessly report continuous glucose monitoring. There's also a company that's making a similar type of contact lens for intraocular pressure. We're doing some work with our school colleagues in engineering to build a series of paper-based sensors. This one can measure 4 analytes and costs fractions of a penny to develop. You just need to put target reagents in the petals of this flower-like looking device, and that might give us the possibility of creating something that looks like a smart tissue. So, when there's diagnostic proteins in the nasal secretions, maybe just blow your nose, it turns blue, and it tells you that you're about to have the flu. So think about that. So then lastly, I just wanted to talk about the big data prospects. And I really loved seeing Mike Snyder a few years ago publish a paper that I'll show you in a minute that had to do with integrated personal omics profiling. So the concept here is that in a future state, but maybe not so far in the future, we'll be able to continuously monitor the changes in our expressed genome, our cytokine profiles, our RNA profiles, our protein profiles. And Mike did the experiment. Mike's from Stanford. He did the experiment on himself and published the results in 2012, where he was able to not only, this is Mike, and these are all the tests that Mike did on himself, which you can't read, but he was able to really follow himself through the course of a series of infectious disease illnesses, through the development of type 2 diabetes. He was able to mitigate his response. He actually wouldn't have known that he was going to get type 2 diabetes unless he was continuously monitoring his glucose, which he wouldn't have done had it not been for this study. So I think this is just a snapshot of what could be done with integrative omics profiling over time, particularly when we have our own baseline as a reference. And the other thing that's happening in this space, besides the omics data, is what's happening, this is Google search data, which I'm sure most of you have seen the notion that Google Analytics allows us to see what kind of search terms people are looking for over time. And this is now a couple of years old, but this is for people that are looking for headache, fever, flu, muscle aches. And you see this spiking pattern over time, which coincides, of course, with the flu season. So the Google Flu Trends analytic data, a big data problem, can actually be used to do some important epidemiology. And at least what was reported in the early phases of this work was this signal from the Google search data preceded the CDC's recognition of the flu season by 2 to 3 weeks. So this data was in the community before medical communities knew about it. And more recently, I'm sure all of you are students of the journal Psychological Science, but these investigators, what they did was they monitored Twitter feeds for language that connoted anger, stress, and hostility, and were able to build a model that showed that the Twitter feeds correlated really nicely with the CDC predicted prevalence of cardiovascular disease across all these counties in the northeastern United States. So, I'm not a psychologist, of course, but I think the kind of language that they were looking for had been validated to be associated with a number of well-defined, psychological sort of predispositions for this. And again, it just shows you what type of data is out there that could be harnessed for better understanding of both population-based disease recognition. I think a few weeks ago, Kristen Newby was here to talk about the baseline study, which is a collaborative longitudinal study between Duke, Stanford, and Google to look at 10,000 individuals. And you can see many of the attributes that I've been talking about, from genomics, the EMR family history, clinical, as well as a wireless device will be used to try to better understand health to disease transitions, particularly for cardiovascular disease and for cancer. And interestingly enough, this is essentially the model upon which the precision medicine initiative that President Obama announced will take shape around the same type of data attributes. Deep genomics, linkage to the electronic medical records, wearable as well as electronic tools, and engaging the patients as partners in this study, not as subjects. I just have a couple of more slides I want to show you, which this is sort of the vision, I guess, of where we could be going, I hope we're going, is where we have a mechanism to capture all this data around us, and importantly, not just capture it, but have the skill sets and analytics to be able to begin to use it not only for the discovery of novel mechanisms of disease, but to reclassify disease and predict what might happen in the future. So speaking of the future, I wanted to show you this brief clip from a company that I have no interest in, Scanadu, but I understand from Bob Harrington that he's now on their board. Whoops, sorry. And this, if all this were to come to fruition, maybe this is what it would look like. Oh, I don't have a microphone. Sorry? Ah, my mic. I forgot to tell you I needed a jack. Do you have one?
I think we do.
Okay. Okay. Do you need to do anything more?
An unprecedented window into the human body.
Up for your volume.
But on a day-to-day basis, we're still in the dark about our own health. We are changing that.
You okay? Do you feel okay?
What if instead of fearing the worst when you notice something out of the ordinary, you could identify the condition yourself? Getting the right diagnosis would save you worry
It says it's roseola.
and an unnecessary doctor's visit.
Rest at home. It's okay.
Instead of hearing about a viral outbreak on the news, imagine you got an alert that was tailored to your family's needs. It would also give you advice about what to do next. What if you had a way to identify what was wrong right away?
It says it's 103.8.
A way to get all of the information you need to understand the situation. And in serious cases, you would know when and where to seek help.
Yeah, it recommends taking it to the urgent care.
We're building a way for people to check their bodies as often as they check their email.
I love that. Check your body as often as you check your email. But and obviously, I'm sure there would be a lot of people that would take issue with this way of practicing medicine. But it's clearly that there are some technologies that will look like this going forward. So I hope I've given you at least a sense of where we are in this journey from having sequenced the genome 10 or so years ago to realization of more of a personalized healthcare platform that utilizes components of these strategies. And I assure you that Duke is one of those places that's going to lead in this area, if not be the leader. But it's going to take all of us, your help, along with many others. And I'm sorry if I've gone a little bit over time, but I'm happy to take some questions. Thanks for listening.
Thank you very much. We have time for maybe one question, maybe two. Go ahead.
So Jeff, that was terrific. And I like your optimism at the end, which I'm not sure I completely share. It seems as though Duke could be doing a lot more, including like with our, $750 million investment in electronic health record for using that for better understanding disease. And and we were told it would take a while for Epic to kind of be usable for these kinds of things. Maybe there are a few little projects that are percolating. But it seems as though we have an incredible opportunity without really having had an institutional commitment to leveraging it. Can you make any comments about that?
Well, maybe my statement at the end was meant to be a little bit provocative to elicit a response like that, so I appreciate that comment. And I don't think that we're in the back of the pack, but if you look at the Emerge network that I presented you a little snippet of, places like Vanderbilt in particular, but some of the other components, Mount Sinai and others, are clearly had an institutional commitment to build an electronic medical record even before we had ours, and also to create the linkages that are important. So, but I think we're also turning the corner, and just my sense, and I don't know if anybody from DHTS or leadership is in the room to validate this, but I do think that the PDC, for example, is beginning to recognize that without the kinds of data repositories of this type and the analytics to go with it, that it's really going to not allow us to practice the most effective medicine, let alone personalized medicine. So I think the investment, some investments will be made. I'm hoping they'll be larger and more substantial than it has been in the past, of course. And people like Mike Pansina here are trying to do exactly that type of thing, to take advantage of our EMR data and to build those kinds of models that we talked about. So hopefully we can bring, people like Mike and his group and other assets in the DCRI to bear on this as well. But it will take a commitment from the top.
Great. Thank you very much, Jeff.
Thank you.